episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Right. Welcome back, everybody, again to another episode of our show with another fascinating individual who is helping to create a better tomorrow for all of us. This time we're headed back to the discussions of healthy longevity. And we have the honor of being joined today by Dr. Tim Peterson, who is assistant professor in the Department of Medicine at Washington University in St. Louis. Dr. Peterson has his PhD in biology from MIT, and he has a lab set up in St. Louis that is interested in a variety of quality of health issues that affect all people indirectly or directly. And the two primary areas of interest that he is focused on especially are an interest in aging, namely health span or the healthy period of one's life, and also mental health and mental health equality for all people. His lab takes a rather broad approach, taking both molecular and population level approaches to identify a range of causal factors underlying these important public health issues. In particular, his lab uses computational approaches, including tools like natural language processing, machine learning, looking at co-expression analysis, as well as areas like wet lab approaches, high-throughput screening, CRISPR screening, metabolomics, and so forth. Dr. Peterson, thank you for taking the time to come on the show and talk to us a little while today.
Thank you so much. Really excited to be here.
It's great to have you. And typically on the show, we start off by handing our guests the floor just for a little while to talk about themselves. If you could sort of take us into the story of who Dr. Tim Peterson is, everything from where you grew up, how you developed your interest in science, in biology, and of course, aging, and a little bit of your career path to date. I think that'd be a great way to start everything off.
Sure. So yeah, I grew up in the Midwest. I'm in St. Louis, Missouri right now. Grew up in Indiana, born in Michigan. So kind of spent a lot of time in the Midwest. And Midwest is kind of derided as the flyover zone. And I think to some extent an uneventful upbringing is not a bad way to be. Can kind of keep you out of trouble. So it's not for everyone, but it was a good place to grow up. So I. Like many people that get into the health world, have some important influences. So my father's a physician, my brother's a physician, my wife and several family members from my wife's family are physicians. So a lot of influences in the medical world. I actually started at medical school at WashU actually, and then about halfway through applied to grad school at MIT. And, because a lot of people do this kind of dual degree, MD, PhD, and where you basically do 2 years of medical school. There's 4 years for medical school, and you do 2 years, and then you go into the lab for about 3 or 4 years, depending how the work goes, and then finish 2 years of med school. So I decided to do that at MIT, and essentially just was so kind of into it that I essentially just didn't complete the medical degree. So I really thought research was kind of what got me excited, and then kind of just took it from there. So just kept going. So but yeah, I mean, in terms of interest in aging and mental health, so these are as you said, these are quality of life issues, and I think a lot of people focus on things that are kind of acutely, cancer, of course, is, acutely can be acutely important. But I think, quality of life issues, situations where you're not going to die tomorrow, but that affect you kind of long term and affect your quality of life. I think those seem kind of underserved areas in the research world, as opposed to some other areas. So, of course, everything's important. Cancer research, as we're seeing with the COVID pandemic, infectious disease, dramatically important. But I think just some areas, just for a variety of reasons, don't get all the attention, particularly in the molecular world, kind of the mechanistic, insight world. So I thought those would be good areas to focus on. And then I think in terms of aging, I mean, actually in terms of both aging and mental health, they're kind of both quagmires. I mean, they're just very hard to think about. Some people think, and I'm in this camp as well, that aging is just kind of there's just kind of this inevitable, a deterioration of our bodies where, you know, it's kind of, there is no kind of underlying principle other than just, it's basically, you can think of like a car falling apart, you know, it's just eventually kind of wears out. And so there's no real pathways or kind of straightforward ways to intervene. And so that, you know, kind of presents an interesting kind of quantitative problem to solve. And mental health too. I think the brain, a lot of people view a lot of people spend a lot of time on the brain just because it's so complicated to think about. And I'm definitely in that camp where, you know, well, is even thinking about a gene really the right level of abstraction to be thinking about? Because if we're talking about a synthesis of neurons and a situation where, you know, everything is just so highly interconnected, you know, how do you kind of put your finger on one spot of that whole network and really expect it to do something that could be helpful to the person. So those are some of the reasons that got me into it. Basically, because they're quite complex, and I think, you know, seems like a good way to, you know, spend a career thinking about. So,
absolutely, definitely.
Yeah. And then, of course, the, you know, the health impact. I mean, of course, as well. You
Sure.
know, the broad health impact. So,
absolutely. You know, you came onto my radar actually a couple of weeks ago. You had this fascinating paper that was in the Journal of Nutrition Healthy Aging. It was titled Moonshots for Aging. And you spend a lot of time in this paper. It's really fascinating, outlining sort of, you know, as you were just saying, we have this aging, very tough problem. It takes a lot of time to study. It's expensive. And you know, we've been doing a lot of things and we don't always question why we do those. And you highlight some of these in terms of, you know, we spend a lot of time looking at the nematodes and the fruit flies and mice. We spend a lot less time, organisms that don't live very long, but we have a lot of organisms that live really long, whether they're naked mole rats or trees or certain types of whales, what have you. We spend less time looking at why. You talk about a major problem for anyone that spent time in the drug industry, that we have this one target, one disease, one pathway, sort of reductionist hypothesis concept the last century in the pharmaceutical industry. And then you hit on something which is incredibly fascinating. It's the last sentence of the paper where you basically say, what would we do to combat aging if we didn't have molecular biology at all? And this is extremely fascinating because I always point out that, you know, there were many fascinating discoveries related to aging before 1950, whatever, when we figured out this thing called DNA. We don't look back, but, you know, we studied aging and biology from more of a physics level in terms of forces and fields and all the weird concepts, which are now coming back a little bit when we realize that cells are great, we're made up of them, but there's a lot more that goes on sort of outside of the cell. Talk us for a little bit through this paper and why you decided to write it.
Yeah, so I think you're kind of, you're touching on a lot of the issues. I think, so just to take that last sentence, you know, what would we do if we didn't have molecular biology? I think, you know, I find molecular biology incredibly exciting. You know, I'm fascinated by the fact that, you know, we have all these genes that, you know, you can't see, but they kind of work together in this kind of microscopic network. And so I think that's, you know, kind of incredibly interesting. I think, though, the downsides it leads to, I'll try not to say this without being kind of too cynical. I mean, there's not really a good way to say it without being too cynical or too kind of, you know, bashing my own, you know, biting the hand that feeds me. But I think it does lead to this, you know, kind of, there's a lot of taxonomy to figure out, you know, gene A to gene B. So you can kind of endlessly play these games about how all these circuits work together. And of course you get, you know, grants and papers depending on, you know, get money and kind of acclaim depending on basically how interesting the, you know, the little network of things that you figure out are. And so, but then ultimately, you know, you're left with, you know, definitely knowledge advance is not trying to discourage my own line of work, but I do think it then, you know, it often doesn't directly translate to something very useful. And so I think it translates to more papers and more grants for the people who do it, but it doesn't translate to kind of the end goal. And often, you know, and so I'm what's called a basic scientist, where you don't necessarily focus on humans, you know, as opposed to clinical research.
Sure.
I mean, we do some of both, but I tend to think of myself as somebody who likes to really, you know, figure out the, you know, the basic facts of how life works. And, you know, And so, and so people in my camp, we kind of have these sentences at the end of all of our papers saying, this has translational implications to human health, and there's no real clear handoff there. And I think most people in my camp are completely unaware of how the pharmaceutical industry works, and the handoff between this gene does this thing to a drug or something that could help somebody. It's just anyone in the pharmaceutical industry knows is a chasm, and so I think there's some issues there. So it's a little bit cliched, but people have said, I think one most famously, people like Steve Jobs have said, having constraints on what you can do kind of can force you in the right direction. They kind of force simplicity, like in terms of like English, if you're a journalist or an author, I think some people argue that the shortest sentence, right, is the best sentence, that it actually takes more time to be concise. And so I think those types of ideas, there's not enough of that. There's kind of like, oh, let's just do things bigger and better. And of course that really works well. You'll see in, Nature, author lists of, 100 people and, the work. There's no way, somebody 10 or 20 years ago could have done the types of works that go in the kind of the big journal papers that come out these days. And so I think it's this kind of quality versus quantity argument, and what are we spending our time doing? And so it's very hard. I mean, so there's a lot of forces. Obviously you can spend, I think there's some Nobel Prize winners who have said, hey, if I was forced to compete today, there's no way I could, because you go 7 years and you accomplish nothing. But so I think there's this kind of pushing the ball down the hill, where it's just easier to do some things. And so we all, including myself, we have to gravitate towards things that can be done rather than kind of what should be done. So I don't know if that really answered your question, but
yeah, no, you hit it right on the target, and that's. I'm glad I understood the paper, and I'm sorry, I'm glad I understood where you were going with it. So they're spot on. So in your research portfolio, you have something called the 10x Lifespan Project, where you you talk about some of the more popular mechanisms of anti-aging research nowadays, and you highlight something like mTOR and sort of a lot of this excitement around things like rapamycin and rapalogs and so forth. Very small lifespan extensions in the model, something like 10%. Yeah. You're moonshotting on this, and you're going for 1,000% or 10x extension. And you talk about in this project, this is going to be discipline agnostic. You're going to look at everything, organismal level, societal, environmental. Talk us through this 10x Lifespan Project.
Yeah, so my background is studying is actually in the mTOR biology, which is a very arguably one of the most kind of hotly studied areas in aging, and actually in a lot of areas of human disease. It's a very important molecular pathway. It controls protein translation, autophagy, mitochondrial function, all you name it, the mTOR pathway controls it. So it's really kind of up in the pantheon of important genes. So it's fascinating. It's amongst the sea of 20,000 protein coding genes, it's certainly very, very, very interesting. And so the issue I have with it, and this is actually not an issue that's specific to mTOR, but is kind of actually starting to get into a criticism of the pharmaceutical industry, which is that, well, we get into these situations where, we have some target pathway, and then we hit that pathway and we get some effect. And all you really want to do, all you really want to do is have a statistically significant effect such that, you can essentially make money off this stuff. And so I think that's where the criticism of the pharmaceutical industry comes in place, because there's just a different incentive there. You just want to get it to work. And it's really like how much it works. Well, that can be debated. And a lot of people, I think his name is Vinay Prasad in the kind of oncology space. He's a pretty active guy on Twitter. He's very critical of his own field, because in oncology, there's all people basically design the metrics. They kind of change the metrics all the time, such that the cancer drug will, extend something, 2 or 3 months, and then, to get approval. And so his critique is that, this doesn't really help the patients, and the drug costs, $500,000 or whatever the drug costs. And I think, obviously some of this stuff's probably exaggerated. I'm certainly not an oncologist or really spent a ton of time in cancer research, but I think the argument is that, well, these companies have to justify their costs. And so there's this huge effort basically to just get this one kind of incremental thing to work. And so in the aging world with mTOR, mTOR is certainly a great target. But I think, the data does speak to, hey, it's 8 to 20 some percent. And that actually is great, right? That's a phenomenal percent increase in lifespan in mice and in a variety of species. I think the downside is that it's not clear, that's what a lot of drugs do on their given phenotype, on a given disease in animal models. And so the problem is when you take it to humans where the studies are less controlled, that 8 to 10%, likely goes away. And so, in the case of rapamycin, just for example, this drug is an immune suppressant, so it particularly knocks down the immune cells the most, and that's helpful because with aging, because your immune system gets hyperactivated. But on the other hand, as you can see with with the COVID pandemic, aging is a huge risk factor for infectious disease. So there's I think just generally speaking, it sounds a little bit dangerous to be putting people in the wild, taking this drug later in life, where they're and chronically taking it, that you'd expect that actually to have a net positive effect. And so, I think studies should be done. Studies with metformin, which is another popular drug which targets the mitochondria. They're being done, and definitely think it's a great idea in dogs and all kinds of species. And this research needs to be done because we do need, these are the gold standard drugs, rapamycin and metformin, and we do need to see, hey, what is the gold standard? And so, I'm probably not the first person that said, okay, let's not try and be incremental about this, or I want to spend time on incremental. And I certainly don't, I'm not delusional to think that we can achieve a 10x lifespan. That's just, that's just not, or at least anytime soon. That's not a really credible thing to say. But I think that having that aspiration is important. And so, I think what makes it particularly tricky is a lot of people believe in this kind of concept. There's this, I think it's it's the old kind of mathematical principle, like the Gompertz law of mortality, where essentially it's a double exponential. The reason that we die, the probability of death is effectively 100% by the time you get to 80 or 90, is because there's not only your kind of the factors working against your exponentially increasing, but they're actually an exponent to the exponent. So the probability of death is, it's hard to overcome an exponential effect, right? But have a double exponential effect is is a tall order. So achieving a 10x effect, is again, it's just the aspiration. So.
Another project that you have in your portfolio is molecular diagnostics for mood, and here, as you pointed out at the beginning, a lot of complex stuff going on inside the skull. You point out that obviously physicians like to measure glucose for diabetes and cholesterol and heart disease, and hormone levels for various other indications. We don't have a rigorous diagnostic tool for understanding our mood that hundreds of thousands of health professionals could utilize. Take us through what you're doing on this front.
Yeah, so we actually started this. This actually led us in a direction that was as pretty unexpected, and actually a little bit away from mental health. So I'll tell you a tangent kind of what we're doing, because we started with this intent, but it's led us, I actually think, in a pretty exciting direction, which we have what's called a preprint out on bioRxiv, and we're going to be releasing a second version of that pretty soon. Cool. So I'll tell you the story. It's actually kind of totally, well, it doesn't really address your question, but I think it takes us in a new area.
That's an interesting story anyway.
So I'll remind me to go back to it. to get into the molecular diagnostics for mood, but I'll just tell you the story to start with, because I think it's pretty interesting.
Cool.
So we started, and it gets at some issues with the drug development world, and et cetera. And so we started, as you mentioned, we do these CRISPR screens. They're quite basic, where you have a drug, you mutagenize cells, you treat those cells with the drug, and then you essentially allow the cells to grow, and you kind of see what grows better or worse. And then at the end of the screen, you get, gene X. You do what's called deep sequencing to basically figure out, which cells, which gene mutation made the cells grow better or worse. So we've done these screens with a variety of drugs. We did them with the antidepressants, the kind of the famous ones. They're called SSRIs, selective serotonin reuptake inhibitors, Prozac, Zoloft. There's a variety of pretty famous ones that a lot of people take. So we did this screen in a cancer cell line, and so right away people were thinking, this is, and I had many people tell me this makes no sense. Why would you ever do a screen to try and understand an antidepressant by watching how cells grow and watching how cancer cells grow? So totally kind of a random kind of experiment. But in yeast and in other model organisms, people have done these kind of experiments to figure out molecular pathways. And it totally kind of, and yeast, of course, has nothing. Many people argue it's nothing like human, but in fact, many of the most many Nobel Prizes can trace their origins to genetics and yeast, and it's in large part, a lot of the most important pathways are conserved, and including the TOR pathway are conserved in yeast, and so we said, there's probably some principles here that we could learn by doing these screens. And then so we not only did those screens with antidepressants, but we used a few other drugs, which all basically have they share some of the same chemical properties. So this gets in a little bit technical, but they're in drug development world, a lot of drugs get designed to basically penetrate the blood-brain barrier. So they tend to make them more lipophilic, and so this has there's kind of ways to make compounds more lipophilic. You kind of add phenyl groups to them, or whatever. And then another thing that's done is they have a reactive amine, which makes them essentially basic. And so there's about 20% of all FDA approved drugs are basically have these two properties. They're decently hydrophobic, and then they have a reactive amine, which makes them basic when they get protonated in the cell. And so these are known as lysoosmotropic drugs. So they trap in kind of lysosome endosomes, or they're also called cationic amphiphils. So they're kind of have anyway, in kind of chemistry, medicinal chemistry world, these compounds are well known. And so the antidepressants are in this category, and essentially when we screen antidepressants and then drugs like chloroquine, which got kind of famous due to the at least kind of infamous, I should say, due to the COVID pandemic. Other drugs such as amiodarone, tamoxifen, which is a breast cancer drug. All these drugs have these what we call cationic empathic properties. And when we screen all these drugs, they all essentially produce the same genetic signature. And that signature largely centered on sphingolipid metabolism. And so most people don't know what sphingolipid metabolism is, but essentially sphingolipids are cationic amphipaths. So they're things that they're lipids that are enriched in certain parts of the cell, which essentially resemble the drug. And so these drugs, the argument is that these drugs, play on this pathway. And you may not know sphingolipids, but they're, just massively important. They don't get the same kind of cachet as cholesterol and maybe some like the unsaturated fatty acids or whatever, but there
was sphingolipid work going on actually down the street at Temple University. That's different talk.
So anyway, so essentially, the kind of the eureka moment and the thing that we're pursuing right now is that all these drugs essentially work not because of whatever is proposed about how they work, but actually the kind of unifying feature of them is that they I mean, they're all chemically different, so they're all slightly do different things, but kind of the underlying pathway there is that they target sphingolipid biology. And so there's a group in terms of the antidepressants, is kind of staying on that thread. There's a guy in Germany named Eric Golbens who really kind of, in terms of the antidepressants, got me onto this idea. So he has a couple of really nice papers where he's essentially showed that the antidepressants work not by targeting serotonin, but rather by targeting sphingolipids. So he's in this camp, and then we've kind of extended this work, and our work kind of goes in quite different directions. But his work has really, over the last decade or so, have made this point quite clear that, you know, at least as far as the antidepressants that all have these cationic empathic character to them, that they all work essentially by targeting sphingolipids. And that, yes, we're not saying that they do not. So the SSRIs, the argument is that they bind to the serotonin transporter in the presynaptic clip and basically prevent serotonin from kind of re-entering the cell and basically keeping it in the synapse so that it can go to the postsynapse and kind of, you know, have its therapeutic effects. Yep. And the argument is not that that's not true. It's just that that's not the dominant therapeutic effect. And so he's done studies actually in worms and in other contexts where you can knock out this serotonin transporter and show that actually that's the mice basically still and the worms basically still have the kind of relevant neurological behavior. So I think that's pretty, I mean, to me, that's pretty exciting because it suggests that, you know, a lot of the, I mean, like I said, 20 or 25% of the drugs on the market, you know, are presumed to work for X, Y, and Z reason. Right. And it argues that potentially they are not working for that reason. And like, there's a recent example in the case of COVID. I mean, the reason why, I mean, this is somewhat of an academic subject, but kind of not, because the reason, like, for example, I'll just tell like one quite recent example. So there's a group at UCSF who has published some. They've been interested in, you know, everyone's been interested in COVID therapeutics, right? And sure. So they basically found in their studies, they found several of these CAD drugs, and several of them are described as sigma receptors binders, and so they've done some studies like other people have shown where if you add these CAD drugs to sigma receptors, that you'll get a very tight binding. So at a molecular level, you say, oh, this is what these drugs do. They bind very, very strongly to these sigma receptors. Unfortunately, when you knock out the sigma receptors in cells or in mice, there really is there's not much of a difference, basically, when you use these CAD drugs. And so I've tried to argue with them that are the journals where they publish their work that these, you know, yes, I'm not trying to dispute that the drugs bind to these receptors. I'm trying to dispute, you know, what's actually conferring the therapeutic effect. And so the reason this is important is you could say, well, the sigma receptors we want to target. So, you know, we've got all these really nice papers that show the sigma receptors is the target, you know, binds to these drugs quite well. So let's just, if we want to make money and, you know, help the world, let's design more drugs to target the sigma receptor. So, you know, all the medicinal chemistry go around, you know, the crystal structures and everything to kind of target that target. But if it's not the right target and sphingolipids are the right target, like it fundamentally changes what the drug development world is should be working on. So, I mean, it's important just because it's, again, where people are spending their time and their resources. So.
Yeah, and I've always been amazed when you open up the patient package insert or the physician package insert on most drugs, how you have that mechanism of action section where it says unknown. And,
yeah. Yeah, and I think you know, and the other thing is that you know, these are often drugs. There's this Lipinski's Rule 5. You know, there's there's rules on how to design a good drug. They're often quite small. You know, I think I kind of feel like, you know, there's often a story that this drug goes into the cell and finds this one protein, and that's can be true, but it's, you know, I think for many of the drugs, particularly the ones that work particularly well, I think they're often, you know, the polypharmacy, the fact that they're kind of ping-ponging around throughout, that's the reason they work. And then the other thing related to that is that, you know, there's this interest in, you know, finding the unique target that no one else is working on that, you know, is kind of some kind of special snowflake that again, telling the story that we're doing something unique. We are the only person working on this kind of pathway.
Yeah.
But the argument against that is, well, most of the drugs that work particularly well are things that target essential processes. Like the example I always like to give, I mean, there's kind of several really kind of famous examples, but the statins, they target cholesterol. They target the rate-limiting enzyme and cholesterol synthesis. I think most people argue those are the most important drugs that exist, one of the most important drugs that exist on Earth. And they target an essential process in all cells. There's arguably nothing more important than that. So it's really just the fact that you're able to not totally deplete cholesterol from the cell when you use those drugs, that's why they work. But you have to start by actually target. I mean, at least my school of thought is that, and many other people that think this way, is that you got to start with something that actually matters.
Yep.
And not some kind of obscure target that you can kind of spin a story about, gene X 1, 2, 3, 4.
Yeah.
Because it's unlikely if it's 2021 and no one's heard of gene 1, 2, 3, 4 to start with, unless you're talking about a monogenic disease where, that's the thing, that's the target. Oh, sorry about that. Where that's the thing, that's the one gene that's mutated in the disease. It's unlikely that gene 1, 2, 3, 4 is going to be, a major player in some common disease like any of the major cancers or any of the, kind of aging related diseases that involve, kind of wholesale kind of changes in physiology. So.
Got it. Working off of that, though, just with the goal of, and I assume this sort of falls under the portfolio disruptive innovation of blockbuster drugs, but would this be, would the goal with the type of research be looking to create better sort of next generation SSRI, or a topic that's kind of hot nowadays is repurposing, taking the SSRI and working in aging instead?
Yeah, I think exactly. So, I mean, and then where this project is going with all these CAD drugs, we're actually kind of the new release of this paper we're going to do, it actually does get into aging, because actually, as it turns out, depression and a lot of these drugs are used as in older adults. And so we're kind of stringing the dots here between sphingolipids and all these kind of commonly used drugs and aging, essentially. And so, I think the idea is repurposing, but I think it could create new molecules. But I mean, I think there hasn't been a great effort to really pinpoint which person should get which drug. And that's coming. I think Regeneron in particular is sequencing a lot of people, and they're going to be tying together all the genetics. I mean, there's a lot of companies doing this, but Regeneron in particular has invested a lot of efforts in sequencing patients. And I mean, I think the challenge there though is, I mean, I think it will be solved this way to some extent. It's not totally clear, but one way in which a lot of people are, with this kind of precision medicine thing that people are thinking is just, you'll have some kind of matrix of genes and, some kind of matrix of drugs, and you'll kind of say, person X will get, this drug, and it'll, they should get it at this level to have its kind of maximally beneficial effects. And so I think hopefully that will get there. One argument though is obviously and so the downsides with, I think, all the particularly useful strategies is they're not going to make anybody any money. I mean, I think repurposing, it's debatable, if you do all that work, how you, and these drugs are off patent, who's going to make money off all this?
Right.
And so I think, that's kind of what drives, things. I mean, I think we'll, in terms of the government grants, we'll do okay. Those are kind of, have been kind of declining relative to inflation over the years. But and then the other major thing is, of course, preventative medicine or preventative, not medicine, preventative, measures, simple things. And this gets into maybe the other parts of this Moonshots for Aging paper, where a lot of basic things that, cost no money, but if they were implemented, I think we would all agree that there could be big effects. Of course, some of the most basic things, like if we all just ate, I think most people would agree if we all just ate healthier, you would see that 10% effect on lifespan, pretty much automatically. I think, other things that I've been pretty interested to see, so like many people have one of these kind of smart watches, and, they keep track of your steps and all that stuff. I think, rigorous testing about whether or not, just simply being up and moving more would again, have some kind of a, 10-year increase type of thing on lifespan, if you're able to implement it widely. And then one other area which I thought was really interesting. So there, I can't remember, was it the Convoy group at Berkeley? But they did something where they basically filtered the blood. The argument is essentially, as you get older, you essentially accumulate too much kind of bad stuff in your blood, and simply filtering that out, had a dramatic kind of younging effect, I think, on mice. And so I think that may or may not be practical to start with, but I mean, one simple thing, there was one paper I saw, that definitely should be more work, this argument that we should all be drinking more water. Well, if you diluted out your blood, would that essentially have the same effect? So drinking water costs nothing. And not saying it would work, but I think it would more likely to work than the millions and billions of dollars it would take to to kind of eke out gene 1 2 3 4, the drug on gene 1 2 3 4. So but unfortunately, saying like, oh, you people take drink eight glasses of water, doing a randomized controlled trial, where that's rigorously tested, it's hard to get that kind of stuff funded. Even metformin, which most people agree is, that trial, I mean, I haven't been too close to it, and there is a trial now, but from what I understood, that trial took a lot of effort to get the $50 million or whatever it was to have people to take it. I mean, it's because there's essentially that $50 million is essentially charity. So I think we do need to kind of return to the scenario where we are, back at least people argue back from, the 40s and 50s, where, money was just kind of given towards, certainly around, just was giving more towards basic research on things which aren't going to yield on the investment right away, but just generally are going to help the world. So I don't know how we get back to that. I think that's a tricky conversation. So
I think that's it's $1 trillion question.
Yeah.
But yeah, you hit it on the head. Tim, what is molecular orphan phenotype matcher project, or Morphiome? What's that all about?
Okay, so yeah, I mean, I think a lot of probably what you're hearing in this conversation, there's a lot of kind of underdog, philosophy.
Sure.
A lot of, hey, let's kind of look at the other side of the coin type of thing. And so I mentioned there's 20,000 genes, and so there's so in my PhD, I characterized one, which the only reason it basically got any attention, I mean, I think it's an important gene, but the only reason it really got attention and enabled us to get some, more grants and more papers is because it functioned in this mTOR pathway. So I think we kind of leverage, it's like we leveraged essentially a kingmaker, and you talk about a gene which no one cares about,
right?
And you tie it to something, that somebody cares about, and there you go. Then it starts to take off as a gene that people care about. So, I find it kind of, is the term like anthropomorphic or whatever, where like genes essentially have kind of life stories, kind of like humans do. And there's of course many famous stories of people, kind of rags to riches, where they are, have somebody that helps them kind of take off in life. And so the inspiration for looking at all 20,000 genes is just that, there's many of them would have a story to tell. Certainly they're not there for no reason. Darwin would argue that. So I think there'd be a value in really trying to understand all 20,000 genes. And so as it turns out, it's really like the same thing with our economic situation, that, roughly about 0.1% of all those 20,000 genes, have all the papers. So it's, over half of the genes essentially have no characterization on them whatsoever. So it really maps quite similarly to the economic situation in our country and in other countries, where most of the genes really, they get no attention, and that, and there's this kind of, if you try and put gene 1, 2, 3, 4 in nature, it's never going to fly unless you tie it to something that people already care about. You know, you can't just say that gene 1, 2, 3, 4 does something. You know, I mean, it's just harder to tell that story unless it has some kind of context, which can kind of catapult it, you know, out of poverty, if you will. And so I've been really interested in that problem. You know, we have essentially, we call it citation inequality. You know, that there's kind of whole areas of biology that just, you know, where there's huge inequalities, and it's a human created problem.
Sure.
And so I think, yeah, so that's the nature of that project. We call it Morphiome, like the orphan, essentially the molecular orphan ome, and ome is this word that people refer to when they refer to kind of a collection of things, in this case, the human genes. So yeah, so I think we're, as part of all our projects, we're kind of trying to, you know, help bring some interesting genes to light that otherwise wouldn't get brought to light. So,
and there's nothing wrong with being the underdog. I mean, there's that whole, you know, universe of possibilities that we, you know, don't step into, whether they be difficult to fold proteins or the odd genes and what have you. You know, we got to do it at some point and figure these things out. So, Tim, this is a wrap-up question coming back to you. Obviously, you know, you've met, worked with a lot of fascinating people over the years as you've gone through your career, developed these projects. Take a little time, if you would, just to highlight anyone that you want, whether they be family members, teachers, other mentors that you want to shout out to that have really been instrumental in Dr. Tim Peterson staying on these paths and moving forward on them.
Yeah, so I mean, I definitely think, so I mean, one of the downsides, I mean, I grew up in even though I had a lot of family members who were doctors and, you know, definitely had all the advantages in life. I think, you know, I think the one thing that's a little bit, you know, speaking to what happens in this country is that I'm not sure everyone really gets, you know, some kind of something that, you know, to become a doctor, you know, it's often just get good grades. You know, it's kind of it's not very romantic or not very kind of inspirational, at least in my view. I mean, I think the helping people part is, you know, it's certainly a huge component of that, but I'm not sure young people get really a chance to do that, you know, I mean, I guess they can, but in my case, I think as soon as I got into lab, and so my grad school mentor, David Sabatini, who really helped pioneer the mTOR field, you know, I think the environment that MIT produces, you know, having that opportunity to do that, you know, which of course I was, again, a lot of privilege to even get in that position, I recognize. But that's, you know, that environment where you get to, your mind gets opened up to science, and that was pretty influential to me. So it took basically, you know, what I think a lot of kids feel, oh, I got to get these grades, or, you know, kind of very mundane things, to, you know, what can I do if I had anything, you know, available to me. And so I definitely speak very highly of the PhD path, because it has that kind of feel to it, you know, where you say to someone, yeah, I mean, you're in some department, so maybe you focus on biology, but I think the nice part about the PhD is that it really allows people to, or any graduate school, is it just allows you to think about what you want to do with your life. And so, and certainly David's, Sabatini's lab fostered that kind of attitude. He wanted to work on things that were important and not necessarily, I mean, yeah, we ended up focusing on mTOR a lot, but I think he would have been open to literally almost anything. And I think that kind of, having that kind of freedom, which I realize most people don't have, I wish more people had that opportunity, because I think that's when you have a little bit of skill and then kind of a clean slate to work on things, you know, I think that's where, and you're young and you have a lot of energy to do it. You know, I think we need to have more of that kind of situation and less of the, you know, sit in class and, you know, less of the, there needs to be a little more unstructured, I think, time for students. So. Yeah, so I think in terms of other mentors, I mean, obviously my parents, I love my parents and brother and family and stuff. You know, that also help. That also is, you know, provides a lot of, I mean, that structure, having that structure there, you know, is also critical. And, you know, to the extent that most people can have that and recognize the value of that and helping people, succeed in life, that's, you know, that's everything. So yeah,
Excellent.
it's yeah, kind of nothing. I'm not saying anything too generic, but it's definitely, I mean, you just, as you get older, you see how, you know, having that structure there, having these people that, you know, can provide those kind of guardrails or mentors to look up to. You know, in the case of my dad, you know, the one thing, you know, that's just overlooked, and I think a lot of successful people will say just basically work hard. And so he, you know, being a doctor, he was a surgeon, he still is a surgeon, you know, just the simple fact that you have to work hard. I mean, it's like, that's not necessarily,
Who think?
I mean, it's very basic, but, you know, I like a lot of successful people who run organizations, and basically it's like, give me somebody who works hard over somebody who's really smart.
Oh, yeah.
So I think that's that basic thing. And, you know, just, you know, it's hard to enrich for just kind of motivation, and it can be brought out in people that don't have motivation. I also think you can bring it out in people, because I think if people lack motivation, that probably reflects, well, they haven't found the thing that they like to do, or they haven't been in a situation which really kind of gets them off the hump. I mean, I was a very motivated person, but again, I didn't really feel like, I mean, I was just kind of jumping through the hoops of getting good grades and stuff like that. And I think when I got to lab and basically kind of abandoned that rat race, and that's where I felt like I really, found what I like to do. So I think it's just, I wish there was more people had more of that kind of opportunity. So.
Gotcha. Right. Cool message.
So.
Good message. Tim, it's a fascinating set of portfolio opportunities you're working on, and, you know, wishing you the best with this. It's really neat stuff. Very diverse. And, you know, obviously, as you said, you know, there's underdogs out there in all these areas, but ultimately they get to where they're going. But as you were saying, keep working and keep pushing all this stuff for us. It's really great. For everybody who's going to be watching this episode or listening across the podcast networks, you've been listening to Dr. Tim Peterson, assistant professor in the Department of Medicine at Washington University, St. Louis. Check out his website. Check out his recent paper, Nutrition and Healthy Aging, entitled Moonshots for Aging. Check out the 10X Lifespan Project, the Diagnostics for Mood Project, Disruptive Innovations of Blockbuster Drugs, really doing a lot. Tim, thank you for taking the time to come on the show out of your schedule. Thank you for everything you're doing. And as we say, thank you for helping to create a better tomorrow for all of us through your work. It's really very inspiring.
Thanks so much, I really appreciate it for having me. And then thanks everyone for listening. Appreciate it.
Absolutely.